Bismillahirrahmanirrahim. Assalamu alaikum. This is Saliha Aziz. The topic I'm going to represent today is ELISA. I am student of Mphil microbiology, Cohort University of Science and Technology. The topic I have prepared is ELISA and I am going to represent it to Dr. Niaz Muhammad. Unlocking the Secrets of ELISA, a journey into the world of immunoassays. Here we have presentation outlines. What is ELISA? How does ELISA work? Types of ELISA, applications of ELISA, advantages and limitations of ELISA, and future of ELISA. Why known as enzyme-linked immunosorbent assay? Antigen or antibody of interest is absorbed onto plastic surface, that's why it is called sorbent. Antigen is recognized by specific antibody, that's why it is immuno. This antibody is recognized by secondary antibody immune, which has attached enzyme linked. Substrate reacts with enzyme to produce product, usually colored. History. The technique was developed in 1971 by Peter Perlman and Eva Engvall at Stockholm University, Sweden. This is a technique which is used to prepare something like immunosorbent to fix antibody to or antigen to the surface of a container, and it was published by White and Jacker in 1966. What is ELISA? Enzyme-linked immunosorbent assay or ELISA is a diagnostic technique and it is used to detect the presence of specific antigen or antibodies in a sample. It is widely used in the field of diagnostic due to its high sensitivity and specificity. The main feature of this test is that it is highly specific and sensitive. ELISA works by using an enzyme-linked antibody to bind to a specific antigen or antibody in a sample. When a substrate is added, the enzyme catalyzes a reaction that produces a detectable signal used as a color change or fluorescence, and this signal can be detected. This signal indicates the presence or absence of the target molecule in the sample. How does ELISA work? ELISA works through a series of steps that involve coating a surface with capture antibody, adding the sample containing the antigen or antibody of interest, washing away any unbound material, and then detecting the bound antigen or antibody using an enzyme linked detection antibody. The enzyme linked detection antibody binds to the captured antigen or antibody, and the enzyme generates a signal when a substrate is added. The signal can be measured using a spectrophotometer or other detection system to determine the concentration of the target molecule in the sample. Here is the working of ELISA. Captured antibody, target antigen is present in the sample. Okay, then we have the enzyme labeled detection antibody, and an enzyme is attached to this antibody where substrate is attached, giving signals in red color. Here are the equipments used in ELISA test. Microwell plate. It is flat bottom polystyrene plate containing 8 to 12 wells holding 350 microliter each. Multi-pipette and 8-channel 100 microliter pipette is a good help for even small scale work. Washing device. It is manually operated and may be of use particularly when there is a risk that sample tested in ELISA contain infectious material, so must be collected for subsequent disinfection. Micro plate washer. These are very efficient with unusually low carryover contamination. Here are the types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. Direct ELISA. Indirect ELISA, the antigen is immobilized on a surface and labeled antibody is used for detection. Antigen is already present. An labeled antibody is used to detect the presence of the antigen. Indirect ELISA and indirect ELISA, an unlabeled primary antibody is used to bind the antigen, followed by labeled secondary antibodies for detection. Two antibodies are used in indirect ELISA. Sandwich ELISA and sandwich ELISA, two antibodies are used to sandwich the antigen between them. Between two antibodies, antigen is sandwiched. Competitive ELISA. Competitive ELISA, the labeled antigen competes with the unlabeled antigen in the sample for binding to a limited amount of capture antibody. Here are the applications of ELISA. As we have already learned that ELISA is really sensitive and specific. So it is used in medical diagnostics, including the detection of infectious diseases, autoimmune disorders, and cancer biomarkers. It is also used in food safety testing to detect allergens and contaminants, and in environmental monitoring to detect pollutants and toxins. For example, ELISA can be used to detect HIV antibodies in blood samples, gluten in food products, and pesticides in water samples. Its versatility and sensitivity make it a valuable tool in a wide range of fields. Advantages of ELISA. One advantage of ELISA is its high sensitivity and specificity, which allows for the detection of very low concentrations of antigens or antibodies in a sample. It is also relatively easy to perform and can be adapted for use in a variety of settings. Here are some, as each test has some advantages, so it must have some limitations. And ELISA also has some limitations. Here we have potential for cross reactivity with other molecules and the need for specialized equipment and reagents. It needs specialized equipment and reagents. It also requires careful optimization to ensure accurate and reproducible results. What is the future of ELISA? How will ELISA be beneficial in the future? The future of ELISA is bright with ongoing research focused on improving its sensitivity, specificity, and speed. There is also interest in developing new applications for ELISA in fields such as drug discovery and personalized medicine. As technology contributes to advance, it is likely that ELISA will remain an important tool in the field of diagnostics and continue to contribute to advances in healthcare and other industries. Here are the precautions while doing ELISA test. First is the use of exchange type pipette. Always use new tip. Washing reagents, use the reservoir for each reagent, label the reservoir. Don't use the same reservoir for multiple reagents. Don't return the reagents to the stock. Plate cover. During incubation, well plate should be covered using the cover plate. Plate cover is effective only under suitable conditions, that is room temperature, humidity less than 50%, air steam 0.2 meter per second. Coating of wells. Coating of wells should be proper with the addition of blocking solution and improper coating. If we have improper coating of our sample or ELISA sample, then we have false positive results. Okay, this is all about ELISA. Thank you so much.